Hey guys, welcome back to my channel. In our last video about cytoskeleton, we talked about microtubules, right? Today we're going to move on to microfilaments and intermediate filaments, okay? So microfilaments is really the smallest one among the three types of cytoskeletons, right? Whereas the microtubule is the largest one. And then the intermediate filament, as the name suggests, is the middle intermediate size one, okay? And then the monomer that makes up microfilament is G-actin. G stands for globular actin. As you can see the shape here of these G-actins, they really have this globular sphere shape, right? And then once you assemble all the G-actins together, we call it the polymer F-actin. F stands for filament, filament actin, or you can call it microfilaments, right? Okay, and then the shape is just like two chains that are intertwined together, okay? But just to remind you, even though this looks like kind of like DNA double helix structure, right? But they're not nucleic acids, right? The G-actins are proteins, okay? And then of course, between the two chains, there's no nitrogenous bases in between them, right? This is a solid structure and they are proteins, not nucleic acids. All right. So the structure of this F-actin is that it has this positive side and negative side, or plus side or minus side, right? So the positive side is where the extension of the G-actin happens, that you polymerize the thing that makes it longer, right? And then when the plus side is doing the extension job, the negative side is usually where it attaches to the cell like an anchor so that the positive side can do its thing, right? And then if you want to depolymerize the microfilament, it also happens at the negative side, okay? All right, so the negative side or negative minus side is the depolymerization, and then the plus or positive side is the polymerization. Okay, so as you can tell, really the assemble or deassemble of microfilaments is really pretty flexible, right? It really assemble or deassembles according to the cell's needs, right? Okay, and then let's talk about the functions of microfilaments. There are three functions you want to know for MCAT. One is to provide tensile strength for the cell, right? So the cell is resistant to compression or fracture or any type of damage, right? So this really sounds like a common function of all three types of cytoskeletons, right? Okay, and then the second one and the third one, the other two functions are really the unique functions of microfilaments, okay? So the second function, which is unique for microfilament, is it's essential. It makes up the essential structure for muscle contraction, okay? I'll make another separate video, dig deep into this. This is really the sliding filament theory, but here I'll just briefly mention that. Look at the structures. This is called actin filament, and the other one is called myosin filament, right? They're also called a thin filament or thick filament, okay? And then these are the structures for muscle contraction. So basically the heads, you see those heads on the myosin, they would bind to the actin filament. And then after the binding, it will literally physically pull the actin filament towards the center of the sarcomere. That's how your muscle contracts, okay? I'll make a separate video about this. All right, so this is the second function of microfilament, which is unique to microfilament, right? And the third one is also unique to microfilament, which is a cytokinesis. And then cytokinesis is a step of what? It's a step of mitosis, right? And of course, meiosis as well, all right? And then before we dive deep into this, let's remind ourselves, what does microtubule do? One of the functions of a microtubule is also to facilitate cell division, but in the phase of a metaphase and anaphase, right? As you can see here, those orange spindle fibers, they're made up of microtubules, right? And then what they do is they split apart the sister chromatids to facilitate cell division, right? During anaphase, right? They pull up, they shorten themselves to pull apart the sister chromatids, okay? So that's microtubules' unique function in terms of a cell division. And then microfilament is also involved in cell division in the phase of a cytokinesis, okay? So how does it work? Is this contractile ring you see here, the material is microfilament, okay? And then the pinch of this contractile ring would give you 2 daughter cells, okay? That's how microfilaments facilitate cell division in the phase of cytokinesis, okay? So now you see how heavily cytoskeletons are involved in the cell division, okay? All right, so this is all about microfilaments, okay? I think we're good to move on to intermediate filament, okay? So intermediate filament, once again, it just has a middle size, okay? It's thicker than the microfilament and thinner than a microtubule, okay? And different than microfilament and microtubule, it doesn't really have a one type of monomer, okay? It has a variety of proteins that are expressed in different types of cells, okay? I'll give you a specific example in just a second, okay? All right, we want to go through the polymers first, okay? So how the monomers of intermediate filaments is assembled, okay? So it's pretty interesting to me that you see that here, two chains of this protein, the polypeptide, they form this dimer, right? And then 2 dimers will fill the tetramer. 2 dimers will assemble as the tetramer. And then these NH2, COO2, these are just indicating that they're proteins, right? The N terminus and C terminus of the protein, okay? And then 2 dimers make up 1 tetramer, okay? And then you see these tetramer, they also have a name called protofilament, okay? So protofilament is really not a new type of cytoskeleton that you need to know, right? It's really just on our way of assemble the intermediate filament that we form this protofilament first, okay? And then a bunch of those protofilament, I believe it's 8 of them, okay? And then 8 of those protofilament, they form this hollow shape. Okay, there's a lumen. See, there's a lumen of intermediate filament, and then they form this hollow shape that is called intermediate filament. Okay, and then I wanna have you pay attention to the. I want to drive your attention to the assembly of microtubules. Remember last time in our last video, we talked about the monomer alpha tubulin or beta tubulin. They're going to make this heterodimer, right? And then a lot of the heterodimers get together. They are called protofilament, okay? So don't get confused. Protofilament is really not a new type of my cytoskeleton, but really the component of our assembly, okay? And then I believe it's 12 of those protofilament also form this hollow shape, okay, where you have a lumen. Then that's the assembled structure of microtubule, okay? All right. Okay. So now we're ready for the example that I said, okay, the monomer, what specific protein is the monomer, okay? It really depends. And then let's see the hair cell, okay? So this hair cell, we have this protein, alpha keratin, okay? And then this alpha keratin, this is an alpha helix, right? Like you see the shape here, alpha helix. And then 2 alpha helixes, they make this, they will make a dimer, and then the dimer will make tetramer, right? And then eventually the tetramer is the protofilament, right? And then a lot of the protofilaments get together, you form this micro intermediate filament, okay? So really, keratin belongs to the intermediate filament protein family, okay? And then there are also alpha helix proteins rich in hydrophobic amino acids. This is super interesting to me because I was super interested in dermatology. And then one thing I wanna interesting fact that's related to MCAT is those alpha helix, alpha keratins, they're really linked. They form this dimer really by disulfide bonds, okay? Disulfide bonds is really high yield on MCAT. And then that's the reason disulfide bonds is really ionic interaction, right? So it's pretty strong intermolecular interaction. So that's why the keratin is a strong thing, right? It's mostly located on the outside of your hair, your nail, your scalp, right? As the protection agents, right? So you really want the protection agents to be really strong. So that's when the disulfide bonds come into play, linking those alpha helix, alpha keratin proteins, okay? All right, I hope that's as interesting to you as to me, okay? All right, So now let's talk about the functions of intermediate filaments, okay? So the first one is intermediate filament also provide tensile strength for the cell, like microfilaments, okay? Like I said, this is a pretty common function of all three types of cytoskeletons, okay? And then the second one is unique, that inner filament is really everywhere. It's located not just in the cytoplasm of the cell, but also in the nucleus. It's located in the nuclear lamina, right? Also supporting the structure of nuclear lamina. And then also even outside of cell, you see intermediate filaments. So intermediate filaments really contributes a lot to the structure, the integrity of the cell, right? So the last one is that, see, the intermediate filament is more of a structural role, not instead of being involved in a lot of the movement functions like filaments and microtubules. Intermediate filament is really more like the spring in the matches as to not the spring to the cell, but cytoskeleton system to the cell, right? Okay, so that's intermediate filament. And then I want to really still remind you, so how are microtubules and microfilaments involved in movements? So these are microtubules, as we discussed in this video, that they literally pull apart, right? They really pull apart the sister chromatids. That's a sort of movement. And then also very importantly, that the microtubules, they are like the tracks in the cell. And then the motor proteins literally walk, not literally, I mean, if you will, they walk on those tracks that are made up of microtubules, and then they transport those motor proteins. They transport organelles or important materials inside the cell, walking on this track that's made up of microtubules. Okay, all right. So that's another type of important movement, right? And then I also remember microtubule. We talked about cilia and flagella, right? I mean, how much movement is that involved? Okay, and then also microfilaments. How do microfilaments move? Literally, those myosin heads, they literally move those actin filaments to facilitate muscle contraction. Okay, so these are the specific functions of microfilaments and a microtubule. They are involved in a lot of movements, whereas this intermediate filament is focusing on the structure, the integrity of the cell. Okay, I hope that's helpful.